ngalakin kadijan yunga boja.
This is the fourth lecture in the series on oxidative stress. So far I've discussed reactive oxygen species, what they are, talked a little bit about some of the terminology associated with it. I then went on and talked to you about this concept of oxidative damage, where reactive oxygen species can damage proteins, lipids, DNA and other macromolecules. I then talked to you about how antioxidant systems can protect cells from reactive oxygen species. So far, I've dealt with the reactive oxygen species in the context of what we might call the prevailing paradigm for oxidative stress. The idea is that reactive oxygen species are detrimental to cell function and they can cause damage. In this fourth lecture, I want to present a slightly different view. And in this lecture, we'll talk about how reactive oxygen species can modify protein function without causing damage to them, and why they can be very important in cell signaling pathways. So this is a little bit counter to this concept that reactive oxygen species are always damaging. Okay, so let's have a look at how this works. This slide shows the concept in a little bit more detail. So in previous lectures, I've been talking to you about oxidative damage by reactive oxygen species. So you might consider the classical view is that reactive oxygen species are considered rapacious, indiscriminate reactive byproducts of cellular metabolism that damage macromolecules. And that's illustrated in this cartoon here. Here we have a number of reactive oxygen species, hydroxy radical, superoxide, you recognize some of them, illustrated as bullies. On the right hand side here we've got our macromolecules such as proteins, lipids and DNA looking particularly fragile, and here we have what might be called the antioxidant cat. So we have superoxide dismutase, catalase, glutathione peroxidase and vitamin E and vitamin C, and basically these act to prevent damage caused by these reactive oxygen species. So that viewpoint has predominated for about the last 40 or 50 years. But in the last decade or so, there's been a bit of a paradigm shift. And the more modern view is that, sure, reactive oxygen species can be damaging, but there is another role for them. And that is that reactive oxygen species can act as signaling molecules which affect signal transduction pathways. In other words, they can have a second messenger role. And there's a couple ways in which they can do that. Here is a classic signal transduction pathway. We have a receptor in a membrane. We have a hormone or some other kind of stimulus that binds that receptor. Once the receptor is activated, it then causes activation of protein kinase cascades, which can then activate transcription factors that then can affect the production of various types of message and ultimately cause the synthesis of new protein. So the way that reactive oxygen species can affect these signaling pathways is firstly that receptor-mediated mechanism could end up generating reactive oxygen species. So NADPH oxidase would be a good example of this. Or reactive oxygen species can affect proteins in these signaling cascades. It could affect the receptor itself, it could affect the protein kinase cascades, or it can affect transcription factors. So what I want to do in this lecture is look at how reactive oxygen species can affect protein function. And the key point here is that the reactive oxygen species are not causing damage to the proteins. So this is a very different concept from the one that's shown on the left hand side where reactive oxygen species are damaging proteins. So let's have a look at how reactive oxygen species can affect signaling transduction pathways. On the right hand side here is some examples of where reactive oxygen species has been shown to affect signaling transduction pathways. Some of these you may already be familiar with, such as NF-kappa beta or perhaps the junk pathways. The point about this slide is that reactive oxygen species can affect multiple different signaling transduction pathways. So it's not just one signaling transduction pathway. So how can reactive oxygen species affect signaling transduction pathways, and what in particular are the targets? Now, reactive oxygen species have been identified to affect signaling transduction pathways in a number of ways. One of them is certainly indirectly via hydroxy radicals, where we have lipid peroxidation products, such as malonyl dihydroaldehyde or hydroxynonenal, that can affect signal transduction pathways. My impression from the literature, this is probably not a major way in which reactive oxygen species affect signaling transduction pathways. Another way that has been identified is through destabilization of iron-sulfur proteins. Iron-sulfur proteins are particularly susceptible to superoxide, and superoxide can destabilize these iron-sulfur proteins and as a consequence affect their activity. Again, I don't think this is a particularly major way in which protein activity is affected. The third way is through thiol group oxidation. In other words, cysteine residues are susceptible to some types of reactive oxygen species, particularly hydrogen peroxide and perhaps peroxynitrite as well. So in this lecture, what we're going to do is we're going to focus on hydrogen peroxide as a signaling molecule and look at some examples. Explain how proteins can be modified by reactive oxygen species without causing them damage, and how these changes in protein activity caused by the cysteine residues can influence cell function. So let's have a look at cysteine residues and why they're sensitive to hydrogen peroxide. Cysteine residues are characterized by this thiol group here, and it's the thiol group that's particularly sensitive to reactive oxygen species such as hydrogen peroxide. It's the only amino acid residue containing a thiol group, and as a consequence it's only cysteine residues that are particularly sensitive to reactive oxygen species. There is another amino acid residue that contains a sulfur, and that's methionine, but that's outside the scope of this discussion. On the right hand side here we've got an example of how this process can work. So here's our protein with a thiol group. Thiol group reacts with hydrogen peroxide to form an intermediate known as a sulfinic acid residue. This sulfinic acid residue is very unstable in biological systems, and the sulfinic acid can react with a thiol group on a small molecule such as glutathione, or it could react with a thiol group on another protein, or in fact even in the same protein. It then forms what's known as a disulfide, either with the protein or with glutathione. So this gives our concept of an undamaging oxidative stress. So the protein exists in one form here and another form over here, and that can change the function depending on whether it's in the disulfide form or whether it's in the thiol form. There is another pathway, so that sulfinic acid group can react with another hydrogen peroxide to form a sulfinic acid residue, and then that hydrogen peroxide can then go on and form what's known as a sulfonic residue. As far as I'm aware, you can consider this irreversible damage. So once the protein reaches this state, that protein, the cysteine residue, the protein will have to be broken down, and the cysteine residue will have to be excreted by the cell because it's no longer functional. With sulfinic acid, there's a little bit of controversy in the literature. Generally speaking, I would consider this to be irreversible, so in other words, the protein is damaged. But there are some cases where there is an enzyme where that oxidized protein can be converted back to the thiol group. But for the purposes of our discussion, we're going to consider both the sulfinic acid form and the sulfonic acid form as irreversibly oxidized. Right, so let's turn to hydrogen peroxide. So we're now looking at hydrogen peroxide as a signaling molecule. So it's got some attributes that tend to make it attractive as a signaling molecule. As a reactive oxygen species, it's relatively not reactive. So it's not really going to react with DNA, for example, or lipids. But it will oxidize thiols on proteins. So in other words, it's reasonably selective. It can cause oxidation of those reactive cysteines. And the other thing I would say is that certainly on intracellular proteins and also in membrane-bound proteins, there are reasonably abundant protein thiols. So in other words, proteins do carry these thiol groups which are sensitive to hydrogen peroxide. Obviously, if there were no thiol groups, the protein wouldn't be sensitive to oxidation. And this last slide down here just gives you the different ways in which the cysteine can be oxidized. So there's our reduced state here. You can resist in the thiol form here, the sulfonic acid form here, which is a single oxidation event. Double oxidation event gives us sulfonic acid residue. Triple oxidation event gives us a sulfonic acid residue. And of course, it can be converted into a disulfide as well. So let's have a closer look at the particular cysteine residues that are involved in regulating protein function. Now, not all cysteine residues are equally sensitive to hydrogen peroxide. And it turns out that the regulatory cysteines are generally located in protein regions where there's positively charged amino acids. The reason for this has got to do with pKa of the thiol group. So the thiol group can exist in two forms. It can exist in a protonated form and it can exist in a deprotonated form. And it's the deprotonated form that's sensitive to hydrogen peroxide. If the thiol group is surrounded by positive charges, that tends to lower the pKa for the thiol group. And so what that means is that at physiological pHs, in other words around about 7.2, you're more likely to have the thiol group in the negative form here, in other words making it sensitive to hydrogen peroxide. This also gives the protein sensitivity and selectivity towards hydrogen peroxide. So the point about this is that not all thiol groups are equally sensitive to hydrogen peroxide. It's predominantly the ones that are surrounded by these positive groups that give it that sensitivity. Now in terms of types of reactions, the different types are shown here. So we've got protein disulfides that can be formed from thiol groups within a protein. So this is an example of intramolecular disulfide formation. And thiol groups can also react with other thiol groups on other proteins to form what's known as an intermolecular disulfide bonds. The thiol groups on proteins can also react with other small molecular weight thiols. Probably the most common reaction, at least within a cell, is with glutathione because it contains a thiol group to also form a disulfide bond, and these are known as mixed disulfides. And finally, I do need to mention nitrosylation. Nitric oxide can react with thiol groups. It's relatively reactive. And then by a series of reactions, which we don't need to understand or look at, we end up with S-nitrosylation of the cysteine residue. And that also has the potential to affect the function of the protein. So this slide summarizes the concept. So in the previous lectures, I was talking to you about oxidative damage. And one of the prime reactive oxygen species that cause damage is the hydroxy radical. So it can cause protein damage, DNA damage, or lipid peroxidation. And the key point about this is it's irreversible. Once you have damage to proteins, the only pathway for that protein is degradation. In contrast, when we're talking about thiol groups on cysteine residues, the process is reversible. So there's the cysteine residue there. The cysteine residue can react with mild reactive oxygen species such as hydrogen peroxide to form these disulfide bonds. That disulfide bond can then affect the activity of the protein. But the process is reversible. So in other words, if there's a reducing environment, the protein can be returned back to its original thiol form. And there's a couple of proteins that can catalyze that reaction. One is thyrodoxin and the other is peroxidoxin. So I want to make sure that you understand that concept. One process is reversible, the other process is irreversible, and it's the reversible processes that we're interested in this lecture. So in terms of reducing disulfide bonds back to thiol groups, I discussed peroxidoxin in the previous lecture. What I'd like to do now is spend a little bit of time talking about thyrodoxin. And the way that thyrodoxin works is shown in this diagram here. So here we have a protein with an oxidized disulfide bond, and thyrodoxin, because it contains cysteine residues, becomes oxidized itself and reduces the protein back to the thiol form. The disulfide in the thyrodoxin can also be reduced back to thiol groups or back to the cysteine residues via an enzyme called thyrodoxin reductase. I showed you this enzyme earlier when I was discussing peroxidoxin. This also involves the oxidation of cysteine residues as well. And the disulfide is reduced back to the thiol groups by a protein-bound cofactor called flavin adenine nucleotide. So FADH2 is oxidized to FAD, and the electrons are picked up from NADPH. So again, we've seen this cycle before. And if this is happening in the cytosol, then it will be the pentose phosphate pathway cycle that's providing the reduced NADPH. Okay, so just a little bit about thyrodoxin itself. It's a relatively small protein. It's around about 12 kilodaltons. It's considered to contain disulfide oxidoreductase activity because essentially what you're looking at here is a series of oxidation of reduction reactions. It's ubiquitous. It's found in both mammalian cells and prokaryote cells. There are several distinct forms of human thyrodoxin. There's a cytosolic form which is found in, which is described as thyrodoxin 1. There's a mitochondrial form which is described as thyrodoxin 2. It contains an N-terminal mitochondrial translational signal. And there's also a variant that's expressed in sperm as well. With the thyrodoxins across the different species, there's a conserved site with cysteine, glycine, proline, and cysteine. So this is essential for function. And thyrodoxin is critical for cell survival. Certainly if the gene is knocked out in mice, it's lethal at the embryonic stage. And one last point I'd like to make with this is that thyrodoxin itself interacts with other proteins. It doesn't directly reduce hydrogen peroxide. Glutaredoxins are another group of thiol sulfide oxidoreductases able to reduce protein disulfides. And the scheme is shown here. We have a protein disulfide here. Glutaredoxin is able to reduce that back to the thiol groups. Glutaredoxin contains cysteine residues, and as part of that oxidation reduction process, we end up with disulfides in the glutaredoxin. The difference here is that instead of using thioredoxin to reduce it back to the thiol groups, we have the cofactor glutathione. So glutathione is oxidized to disulfide, and that oxidized glutathione can then be reduced back to the reduced form by the enzyme glutathione reductase using NADPH as the source of electrons. One specific aspect of glutaredoxin is worth noting is that it can also reduce mixed disulfides. So mixed disulfide is where the thiol group on the protein is reacted with a small molecular weight thiol group, a compound such as cysteine or glutathione. And in this example here, you have a protein with a glutathione residue on it. In the presence of glutaredoxin, we have a thiol exchange reaction where the glutathione is transferred onto the thiol group of the glutaredoxin to form a mixed disulfide, and then that disulfide is reduced by glutathione, so the cofactor glutathione. So this distinguishes it from the thioredoxin, which can only reduce protein disulfides. So if we compare thioredoxin and glutaredoxin, they certainly have overlapping functions, so they can both reduce protein disulfides. They do have a common motif with a cysteine, N-amino acid, which is what the X stands for, and cysteine. And the difference is that glutaredoxin is reduced by glutathione, whereas thioredoxin is reduced by thioredoxin reductase. And the other difference is that glutaredoxin is capable of reducing these mixed protein disulfides. So the next question we can ask is, what proteins can be affected by thiol oxidation? It turns out there are a range of different types of proteins in many different pathways which can be oxidized. So the thiol groups can be oxidized and their function can be affected. And that's what this diagram shows here. So using cell culture experiments and looking for proteins that are affected by oxidative stress, it's been discovered that kinases and phosphatases in signal transduction networks are affected by thiol oxidation. Mitochondrial proteins can be affected. DNA proteins, DNA repair proteins can be affected. Metabolic proteins, so in metabolic pathways such as glycolysis, can be affected. Protein translation can be affected. A variety of antioxidants themselves are also sensitive to thiol oxidation as well. Endoplasmic reticulum, contractility, and so on. So really the point about this is that we know from cell culture studies is that there are a large range of proteins sensitive to oxidative stress. Now I should point out that this is in cell culture studies. We don't necessarily know whether these proteins are also affected in disease conditions where we're seeing oxidative stress. So a little bit more work needs to be done for the in vivo environment. Now, oxidation of these proteins can have effects on different cells and cause different cellular outcomes. So, for example, one cellular outcome is that with low levels of reactive oxygen species causing thiol oxidation, you can have increased in proliferation. Certainly looks to be important in differentiation. Thiol oxidation can lead to cell death by apoptosis and also necrosis by these thiol oxidation methods, and we've got effects on protein turnover and also muscle function. How a cell responds to something like hydrogen peroxide is really going to be dependent on the nature of antioxidants it has, the type of cell we're dealing with, and also the concentration of hydrogen peroxide. And this slide shows you how cells can respond to hydrogen peroxide, to different concentrations of hydrogen peroxide. So this is from a cell culture experiment. Don't assume necessarily that the concentrations that you look at apply across all cell types, but it's just meant to give you a bit of a guide. So firstly, when we're looking at hydrogen peroxide, certainly if we're doing it in cell culture, the extracellular concentration you're applying doesn't necessarily equate to intracellular concentration of peroxide. So peroxide can cross membranes, but once it enters the cell, it can be broken down, and that means that the intracellular concentrations are not necessarily equivalent to extracellular concentrations. However, looking at this, if we take a concentration in the range of 0.1 to 7 micromolar peroxide, we can get stimulation of proliferation, which is what's shown here. That occurs over a range of peroxide concentrations. As the peroxide concentration keeps increasing, we can have temporary growth arrest. In this scenario, we have the synthesis of stress genes, or the expression of stress genes or heat shock proteins. So in other words, the cell can adapt to that stress. That means that these cells may then continue proliferating or growing depending on what they're doing. As the concentration of hydrogen peroxide keeps increasing, we can have permanent growth arrest, but the cells still survive. Keep pushing up the hydrogen peroxide concentration, then you move into a region where you have cell death. Typically what you see is initially you see programmed cell death, so cell death by apoptosis, but if the levels are high enough, you end up with cells dying by necrosis. So the point about this is how the cell's response is going to depend on the level of peroxide stress that it's exposed to. So in other words, different proteins are going to be sensitive to different levels of hydrogen peroxide. So I've been describing the response of the cell in generic terms. What I'd like to do now is drill down and show you two specific examples of proteins that respond to oxidative stress. And my particular example is going to be around apoptosis. So I've said to you that peroxide can cause cell death by apoptosis. So how can that happen? Well, there are several different pathways by which it can happen. I'm going to show you two proteins that are likely responsible for it. One is MEKK1 and the other is ASK1. So this diagram shows you some of the signal transduction pathways that are responsible for cells responding to different types of extracellular signals. The one that we're interested in here is oxidative stress. But really what this is showing you is a receptor-based system which responds to a signal that then can affect a group of proteins called kinase. Those kinase can then phosphorylate a group of proteins called kinases, or MKK for short. And then we have the phosphorylation of a group of proteins called mitogen activated protein kinases, or MPKA for short, such as JUNCs and so on. And these are transcription factors that then can give some kind of biological response at the transcriptional level to that particular signal. The one that we're particularly interested in is this oxidative stress one. And in this particular diagram, it's focused on ASK1, but we've also got effects on MEKK1 as well. Let's start with MEKK1. So what you're looking at here is the domain architecture of MEKK1. MEKK1 contains a kinase site. That kinase site is responsible for causing phosphorylation of protein substrates. For that to happen, ATP needs to bind to a site on the kinases. In that ATP binding site, there's a cysteine residue, or cysteine 1238 in the binding site. If a mixed disulfide bond forms, so in other words that thiol group is sensitive to oxidation, if we get the formation of a mixed disulfide, then that interferes with the ability of ATP to bind. Now generally MEKK1 is thought as a cell survival factor. So when we're looking at signal transduction pathways, we can divide them into cell survival factors or cell death factors, and whichever one predominates is going to affect the outcome for cell survival. So MEKK1 fits into the cell survival factors. Now generally speaking, MEKK1 would go ahead and phosphorylate its protein substrates. That would lead to gene expression and then the production of proteins that were involved in protecting the cell or keeping the cell alive. What happens in an oxidative stress event is because that cysteine residue is oxidized, MEKK1 can no longer phosphorylate its protein substrates, which means that you lose a cell survival signal. And that tends to tip the balance towards cell death by apoptosis. Another protein in the cell signaling cascade is ASK1. Now, unlike MEKK1, ASK1 is considered to be a cell death protein. So in other words, it can promote cell death. So it's involved in various functions. The one that we're particularly interested in is apoptosis. The way this works is a little bit more indirect than with MEKK1. So ASK1 normally exists bound to thyrodoxin. And the thiol groups on thyrodoxin are important for the binding to ASK1. So this is a non-covalent interaction, by the way. And that normally triggers ubiquination and degradation. So generally speaking, if ASK1 is binding to thyrodoxin, its pathway will be a degradation pathway. In the presence of reactive oxygen species such as hydrogen peroxide or cell death signals such as tumor necrosis factor, what happens is the thyrodoxin is oxidized and we form an intramolecular disulfide bond. That decreases the affinity of thyrodoxin to ASK1, so the thyrodoxin detaches, and that then allows the ASK1 to form a dimer. In the dimeric form, ASK1 is now activated and it's a cell signal for promoting apoptosis. So the cell then goes on to die by apoptosis. So we know from many in vitro experiments, in other words cell culture experiments, that reversible modification of proteins by thiol oxidation can affect their function. And I've given you two specific examples, one with MEKK1, which is a cell survival signal, and the other with ASK1, which is a cell death signal. But there is a bit of a problem. So most of the thiol oxidation events that I've been talking about have been for model systems, and also with chemicals that generate a strongly oxidizing environment. This issue was raised quite a few years ago, and the point being is that in many cases we don't know whether these events are actually happening in vivo. So are they actually happening in disease? So there still has been very little work that's actually been done in animal models of disease or in humans demonstrating that oxidation of thiol groups is actually contributing to the pathology of diseases. So certainly one of the things that we need to find out is that we need to find more evidence that protein thiol group modification is actually occurring in vivo. In other words, in the tissue of animal models and humans. And to discuss that, I want to give you some examples from our research. So our research group has been discussing this problem. Can we show that protein thiol oxidation is actually occurring in tissue? Some time ago, we developed a fluorescent labeling technique to find out. I won't go into the details of the technique, but if you're interested, there's a publication that you can have a look at. But in essence, it involves attaching fluorescent dyes to both the oxidized and the reduced thiol groups. And so that's what's shown here. So we have our thiol groups here. We bind the reduced ones with tag 1, and then we bind the oxidized thiol groups with a second tag, which is shown in red here. These have different excitation emission wavelengths, so we can pick up what's actually happening to the oxidation state of the tissue by measuring the relative concentration. So if we have increased oxidation, for example, we're going to have increased fluorescent tag number 2. And so that means that we can look in tissue, and we've looked in muscle tissue, to work out how much protein thiol oxidation is occurring. And for that, we can just take the level of oxidized protein, so tag 2, over the total number of thiol groups in the tissue. Now, we are interested in various disease models, but most of our work has been done in Duchenne muscular dystrophy. This is a genetic disease affecting about 1 in every 3,500 to 6,000 boys worldwide. Ultimately, it's a lethal disease, and it's caused by a dysfunctional protein called dystrophin. Duchenne muscular dystrophy is associated with oxidative stress. So the question has been, is oxidative stress contributing to the pathology of muscular dystrophy? And the pathology involves muscle wasting. So what happens with these boys is that over time they lose their muscles. You have contraction, which is what you can see here. They lose the ability to walk, and eventually they die through respiratory failure. Now, we've been looking at this in a mouse model of muscular dystrophy. It's missing the dystrophin protein, such equivalent to what's happening in the boys, and it's referred to as the MDX mouse model. So we've looked in this model to see if we can find protein thiol oxidation. So with the MDX mouse model, if we look at a timeline of what's actually happening to the muscles, once the mouse is born, at about 3 weeks, we begin to see increasing levels of necrosis and inflammation, and we also see increasing levels of oxidative stress as well. This severe bout of necrosis inflammation continues for about another 3 weeks, and then gradually decreases, and by 8 weeks there's very little necrosis and inflammation going on. So what we were interested in finding out is whether there was protein thiol oxidation in these muscle samples. So we used the technique I described earlier to measure protein thiol oxidation. The first thing is that we looked in different muscles, so quadriceps, triceps, gastrocnemius and diaphragm, to find out whether there were changes in total protein thiols. And in red is our dystrophic mouse, in blue is our control mice. And you can see that there's no real variation between MDX and control mice, although there are variations in the total amount of protein thiols between different muscles. When we looked at the degree of oxidation, that's when we started seeing differences. So you can see that in the quadriceps mouse muscle, there's quite a substantial increase in protein thiol oxidation. And we also saw increases in protein thiol oxidation in gastrocnemius and also in the diaphragm as well. One of the muscles that we didn't see protein thiol oxidation occurring was the triceps muscle. And this is quite interesting because the triceps muscle is not very severely affected compared with the quadriceps, gastrocnemius and diaphragm. And so really what this is saying to us is that there is potential for protein thiol oxidation to be contributing to the pathology of the dystrophy. Now, we haven't proven it, but this is the first step in showing that relationship. We've also used microscopy to look for protein thiol oxidation as well. And this example here, you're looking at cross sections of mouse muscle, and we've used a stain to have a look at what's happening with that mouse muscle here. So here you can see healthy muscle fibers, but you can also see disrupted muscle fibers as well. So there's one patch of disrupted muscle fibers here, so we're looking at necrosis occurring here, and there's another patch over here. We developed our fluorescent technique to look for protein thiol oxidation in these muscle fibers. And the way it works is that the lighter it is, the more oxidation that's occurring. And you can see that where we have disrupted fibers, in other words, fibers dying of necrosis, we also have very high levels of thiol oxidation. So in this patch here and this patch over here. So again, this supports the idea that protein thiol oxidation is contributing to the pathology that we see in necrosis. And of course, this is interesting because if oxidative stress is contributing to the pathology, then perhaps we can use an antioxidant to prevent thiol oxidation and then protect the muscle from the pathology. Now, this brings me to one last point, and that's antioxidant therapies. You'll often see in the media claims about antioxidants being used to protect people from various chronic disease states. However, if you look at the clinical trials, the outcomes have been very poor in using antioxidant supplements, and this just gives you a few here. So in this review here, there was no evidence to support that antioxidant supplements prevent mortality in healthy people or patients with various diseases. Antioxidants in diabetes, dramatic failure of clinical trials with antioxidants. One of the problems with oxidative stress is it's thought to contribute to the loss of vision. Again, current evidence does not support a protective role for dietary antioxidant vitamins. And then in cardiovascular diseases such as atherosclerosis-related cardiovascular disease, again, supplements with vitamin E or vitamin C do not provide a general benefit for protection. In our area of research, so looking at muscles in particular, increased antioxidant supplementation did not prevent muscle atrophy during immobilization. So one of the questions you can then ask is, well, why have all these clinical trials failed? Is there something that we don't understand? And I would argue, yes, there is something that we don't understand. We don't understand how oxidants are really affecting tissues. So this slide summarizes that conundrum, and it maybe explains that conundrum. And I would argue that, in fact, protein thiol oxidation is where we should be looking for in terms of our antioxidant therapies. So let's have a look at the pathway. So here we have oxygen picking up an electron, superoxide, so generation either deliberately or inadvertently. We have the formation of hydrogen peroxide. Then with the Fenton reaction, we have the potential for the formation of hydroxy radicals. And those hydroxy radicals can then cause irreversible damage to macromolecules, and if it's severe enough, we can affect cell function or cause cell death. Now, if you look in all of the clinical trials, what they've been using is chain breaker antioxidants such as vitamin E and vitamin C, or they've been using things like ion chelators. And certainly, those compounds would help protect cells if, in fact, hydroxy radicals were involved in this pathway that's shown here. But perhaps that's not how it's working. Perhaps what's happening is that hydrogen peroxide is affecting proteins through thiol oxidation. And it's those proteins that, when become oxidized, that then cause cell dysfunction or even cell death. The problem is, if we look at the antioxidants that have been used in clinical trials, they're all tackling this hydroxyl pathway. They're not tackling this hydrogen peroxide pathways. And so the reason that clinical trials might have been disappointed is because the focus has been wrong. The focus has been too much on the damage pathway and not enough on thiol oxidation. That's certainly one possibility, and that's something that we are following up on. Okay, so that completes this lecture. So in this lecture, I've really focused more or less on cell signaling, so on thiol oxidation primarily, and the role of redox active cysteines in that response to reactive oxygen species. And the primary ROS that I've brought to your attention here is hydrogen peroxide. It's not the only one, but it's thought to be a major player. I've discussed protein modification, the types of modification that you can get. So you can get protein disulfides, either intramolecular or intermolecular. We can get mixed disulfides, and I've also discussed nitrosylation as a mechanism for causing cysteine oxidation as well. Importantly, I've indicated to you that this process is reversible, and so we can distinguish it from the prevailing concept is that oxidative stress always causes oxidative damage. And we've got a couple of proteins that can reverse protein thiol oxidation, so thioredoxin and glutaredoxin are examples. And then I finished off by giving you some specific examples about how protein thiol oxidation can affect cell function. And then finally, I've talked about how we need to make protein thiol oxidation more relevant to in vivo scenarios. So we need to better understand how that thiol oxidation is in fact affecting tissue pathology in disease models. So I hope you found that interesting and you've got an opportunity to ask me questions in the review session.